Hello everyone and welcome to another recorded lecture of Anatomy and Physiology 1 online. Today we will discuss Chapter 5, which is on histology, the study of human tissues. So what is a tissue? When we're discussing tissues, we're not talking about the ones you have on your desk right now. We're talking about a group of similar cells that all work together to perform a similar function. So the definition of a tissue is a group of similar cells and also their cell products that work together to perform a specific structural or physiological role in an organ. These cells of a tissue are embedded in a matrix of proteins and fluids. Sometimes it's not necessarily a fluid, but more of a gel or a rubbery substance. But tissues, we're going to see, are just cells embedded in stuff. That's what a tissue is. There are four basic types of tissues that we'll discuss. The first are epithelial tissues. And epithelial tissue is composed of closely spaced cells that cover the surfaces of organs and are really important for protection, secretion, and absorption, as we'll soon see. Connective tissue binds and supports organs, also has a protective function as well. Muscle tissue is meant for contraction to provide movement. And finally, nervous tissue can process information because it contains excitable cells that are specialized for rapid transmission of coded information. We're going to talk a lot more about muscle tissue and nervous tissue in subsequent chapters. So we're going to focus mostly on epithelial tissue and connective tissue. If you were in an anatomy and physiology class, you would be looking at slides quite a bit. And these slides on a microscope have to be prepared very specifically so we can visualize these tissues. So to visualize cells and tissues on slides, we must fix and embed specimens. Fixation means we first take a small piece of tissue. Let's say we want to look at the liver. So we're going to take a small piece of liver and we put it in a fixative solution like formaldehyde or formalin. And what that does is it cross-links all the proteins in that tissue, meaning it's going to freeze them in place. So this gives us a snapshot of what's going on inside that tissue. That formaldehyde can also inactivate any degradative enzymes that would normally break down the cell structures. So by inhibiting those enzymes, we're going to preserve all the cell structures in the tissue. So after we fix our tissues in a fixative, we have to dehydrate them. So the fixed pieces are soaked in increasingly more concentrated alcohol solutions. This will remove all the water from the tissue. If there's water, that means the tissue can decompose, right? And we don't want it to be contaminated with anything. We want it to be completely dehydrated so it's stable. And we can then remove all the alcohol by putting it in a clearing solution. Finally, we have to embed and infiltrate. What that means is we take the tissue and we put it in melted paraffin wax. And that wax will infiltrate it and allow the tissue to harden. So now so imagine like a piece of tissue that's in paraffin wax, and then it allows it to harden in a paraffin block. You can then do sectioning and mounting, which is you take the paraffin block and you slice it in very, very, very thin slices, about 1 to 2 cells thick. And you then mount them onto glass slides before you stain them. Right? And staining is very important for contrast. So H&E stain is commonly used in histology. Hematoxylin, so H&E has 2 different stains in it. Hematoxylin is a basic dye that can stain acidic structures, purple or blue. So since DNA and RNA are nucleic acids, they're acidic. Nuclei and ribosomes are stained purple by the hematoxylin in the H&E stain. So you can see over here, all of these cells, these nucleic acids are stained darker with H&E. Eosin is an acidic dye that can stain basic structures pink. And most proteins in the cytoplasm are basic. So the cytoplasms are stained pink in the image below by the eosin in the H&E stain. So sectioning a tissue reduces a 3-dimensional structure to a 2-dimensional slice. So it's important to keep in mind how tissues can be sectioned. So when you're looking at a specimen, you can try to relate it to the actual 3D structure in the human body. So you might see a longitudinal section, LS, which is how the tissue is cut at the long axis, making like a frontal plane cut. A cross section you can look at, that's a transverse section. So usually on a slide it will say CS or XS or TS for longitudinal section. Right, that's when you want to cut perpendicular to the long axis. Or you could also have an oblique section, which is where you want to cut the tissue at an angle between a cross and a longitudinal section. So these are all going to be specified on a glass slide before you visualize it on a microscope. So before we talk about tissues, it's important to realize that all tissues derive from 3 primary germ layers. So what does that mean? When sperm and egg meet, they form a zygote. So a fertilized egg is called a zygote. And as that zygote divides by mitosis, divides, the embryo splits up into 3 primary germ layers. Those 3 primary germ layers will give rise to all the structures in the fully formed adult. The ectoderm, or the outer portion, will form the epidermis and the nervous system. So this portion will form the epidermis and the nervous system. That's the ectoderm. The endoderm will form the lining of the gut, the digestive organs, and the lungs. So the digestive tract, digestive organs, and the lungs are all from the endoderm. You can remember endo from inside, so they're inside your gut. Mesoderm is the middle, and that's what gives rise to cartilage, bone, and blood. So all connective tissues. Also muscle, it comes from mesoderm. So that's just very important to keep in mind that after this phase in development, all of these cells will continue to differentiate, but these yellow cells only know how to become gut and digestive organs, and blue only knows how to become nervous system and skin. Mesoderm only knows how to become muscle and connective tissue. So now we're going to go through different kinds of tissue one by one, starting with epithelial tissues. So epithelial tissues are distinguished by 5 characteristics. They all have closely packed cells. And that's the first thing that they all have in common. They also are all attached to underlying connective tissue by a basement membrane. So here's the underlying connective tissue, but there's a basement membrane that kind of anchors all the cells to the basement membrane, and that's what separates the epithelial cells from the connective tissue underneath it. The tissue opposite the basement membrane is not attached to any other tissues. That means this is the free surface. There's nothing right over here. Right? By definition, epithelial cells have one end attached to a basement membrane and a free end as well. This is called the apical surface compared to the basal surface. So the apical surface is the free surface exposed to the external environment. Epithelia are avascular, means that they do not have any blood vessels, but they do lie on very vessel-rich connective tissue, which can furnish them with all the nutrients that they need. The tissues regenerate very rapidly. So they actually repair themselves very quickly. They can do mitosis very efficiently. And this is important because skin is very vulnerable to injuries. So we need them to regenerate very rapidly. So epithelial tissue has several functions that we must understand before moving forward. So protection. Epithelia are responsible for protecting deeper tissues from invasion and injury. So for example, the epidermis of the skin is a barrier to infection, and the inner lining of the stomach protects its deeper tissues from stomach acid. So epithelial tissues are good for protection. They're also good for secretion and excretion. So secretion is how epithelial produce mucus, enzymes, hormones. So glands, which secrete all these things, are mostly composed of epithelial tissue. Excretion is very related as well because epithelial could get rid of all the waste from tissues. In the lungs, we have to get rid of any excess CO2 to exhale it. So that CO2 can actually be excreted across the pulmonary epithelium, so then we could release it to the external environment. Epithelial tissue helps with absorption. For example, in the small intestine, our epithelium can absorb all of our nutrients from the food that we eat, so it can get into our bloodstream. There's also a diffusion and filtration function of epithelial tissue. So substances that leave the blood can be filtered through the epithelium that lines the blood vessels. It's called mesothelium. And basically this allows only certain things to get in or out. So epithelial tissues allows for certain things to get in and out, which is diffusion or filtration. And finally, there's actually a sensation role, right? So epithelial tissues help us sense things because epithelia are provided with nerve endings that can sense stimulation from a touch to irritation to heat. So functions are very widespread in epithelial tissues. So let's talk about simple epithelium first. Simple epithelium is tissue that has one layer of cells. So simple means one layer, right? Single layer of cells. The cells may be squamous, may be flat-shaped. They may be cuboidal, almost like cube-shaped, or they may be columnar, which are column-like. Simple epithelium is important in places where rapid diffusion, secretion, or filtration occur. So we want only one layer of cells when we want things to pass through quickly, right? That makes sense. We said that there's a basement membrane that separates epithelial cells from their underlying connective tissue. This basement membrane has a lot of proteins and carbohydrates. And again, it kind of anchors the epithelium to the connective tissue and controls the exchange of materials between the epithelium and the underlying tissues. So now let's talk about different types of simple epithelium. We said simple epithelium only has one layer of cells, but it can be simple squamous, simple columnar, or simple cuboidal, or there's actually another type, a fourth type we'll talk about. So the simplest type of simple epithelium is simple squamous epithelium. It's a single layer of squamous cells. So these are really important in osmosis, diffusion, but for also friction reduction. And we'll talk about that more when we talk about membranes. Simple squamous epithelium forms the air sacs of lungs. It forms the mesothelium. The mesothelium protects our internal organs. And our endothelium lines the insides of our blood vessels. So mesothelium protects our internal organs, regulates things that go in and out. Endothelium lines the internal lining of our blood vessels and allows in and out. So we need rapid diffusion of gases and nutrients. So that's why we only want to have a very thin layer of the thinnest cells possible so things can go in and out very, very easily. So mesothelium is a type of simple squamous epithelium that protects the organs. Endothelium is a type of simple squamous epithelium that lines the inside of blood vessels. And remember that the free surface is called the apical, and the end that's connected to the basement membrane is called the basal end, or the basal surface. So now we'll talk about simple cuboidal epithelium. So it's simple, so we still have a single layer, but they're cuboidal cells this time, right? So they're single layers of square or round cells. We see these when we need absorption and secretion. The representative location is the kidney tubules. You'll see cuboidal cells in the kidney tubules, but they're also found in the thyroid, liver, and in other glands. So liver, thyroid, glands, and kidneys, you should remember, is where you're going to find simple cuboidal epithelium. So again, this is very important for absorption and secretion in the kidneys. It also helps with moving some mucus around. So that's simple cuboidal epithelium. Next up, we have simple columnar epithelium. And in simple columnar epithelium, we have a single layer of columnar cells, right? They're very narrow. And these are aiding in absorption and secretion as well, but also for protection. We see these lining the internal surfaces of our stomach and the intestines, also the uterus. So we see absorption, but also secretion of mucus because they contain goblet cells. These are goblet cells over here because they look like a goblet. Right, so you can see here these columnar cells, columnar, single layer. So that's why we know that it's a simple columnar. But they have these goblet cells that produce mucus. Note these are the nuclei. They're all in one layer. Right, so those are the simple columnar epithelium. Next, we have pseudostratified ciliated columnar epithelium. So this is a little trickier because in pseudostratified ciliated columnar epithelium, it's still a single layer. So it's still simple epithelium, but it visually looks like many layers. Right, so when you look at this pseudostratified ciliated columnar epithelium, it looks like the nuclei are staggered at different positions. But in reality, this is still one cell thick. The nuclei are just at different positions. Almost like trees in a forest look like they have many different layers, but they're all anchored to the same ground, right? The trees are all at the same level, but it looks like many levels because their branches extend to different heights. Pseudostratified ciliated columnar epithelium always has goblet cells to produce mucus and, as the name suggests, cilia. And remember that cilia are those short little hair-like protrusions made of microtubules that can brush and propel mucus and any entrapped particles away from anything important and toward the digestive system so we can digest any of those pathogens. So pseudostratified ciliated epithelium lines the upper respiratory passageways. Right, this is very important. So our upper respiratory passageways are exposed to dust and pollen and pathogens. So what we need to do is we need to make mucus. Right, that's where the goblet cells come in. But also, once we make the mucus to entrap the dust, we want to use that cilia to push it away, push it down the nasal passage, right, away from anything important like the brain. So it could go down to the stomach. It's different. So pseudostratified ciliated columnar epithelium is unlike simple columnar because it's not absorbing anything. Right? This is more protective. This is getting things away. It's pushing them to protect us. They're not absorbing nutrients like the columnar epithelium of the intestines. So that's a big difference between the two. So that was the simple epithelium. Those are single layers of cells. But now let's talk about stratified epithelium, which consists of more than one layer of cells. So stratified epithelia range from 2 to 20 or more layers of cells. Some cells rest directly on others, where the deepest layer only are attached to the basement membrane. That deepest layer attached to the basement membrane is called the basal layer. And that's where the division occurs. That's where mitosis occurs in the basal layer. So that's very, very important to remember. Mitosis does not occur anywhere above that first most basal layer. The superficial cells meaning the ones closer to the external environment, are continually lost. So cells grow at the bottom and are lost from the top. And these are really important for protection, right? We need multiple layers because we need multiple layers of protection. Right? The epithelium type is determined by the cells at the free surface. So that's important. So the deeper cells may be a different shape than the surface cells. So when we talk about the stratified epithelia, we're naming them based on the shapes of their surface cells. So they could be stratified squamous, stratified cuboidal, or stratified columnar. We're going to really only focus on stratified squamous in this class. So stratified squamous epithelium has cells on the free surface that are squamous in shape, right? Those flat cells. These are important to provide resistance to abrasion. So again, protective. We need multiple layers for protection. Keratinized cells form the epidermis. So in the epidermis, we have many layers of keratinized cells. What is keratin? Keratin is a waterproofing protein. that gives extra protective mechanisms to these cells. So cells continue to fill themselves with this waterproofing protein called keratin. It's a type of intermediate filament protein. And that's what forms the epidermis. So on the skin on the outside of your body, it's called a cutaneous membrane. That's composed of stratified squamous epithelium that is keratinized. We have non-keratinized stratified squamous epithelium as well. We see those more on the tongue, the mouth and tongue, and the esophagus, and the lining of external genitalia. So whereas we see keratinized on our epidermis, so like the soles of our feet and the palms of our hand have tons of keratin, so we can resist abrasion. It also protects us from water loss because it's like a waterproofing protein. The non-keratinized type is exposed to liquid, so we don't want keratin, right? So when you think about these tissues, they're in aqueous environment, so we don't want them keratinized. We don't want them waterproofed, right? But both, regardless, are for protection. We need many layers for protection. Stratified cuboidal epithelium is very rare. We see those in glands, in the sweat gland ducts, and sperm-producing ducts of the testes. Stratified columnar epithelium, again, it's pretty rare. Kind of resembles stratified squamous epithelium, but we have columnar cells. And these are found in the male urethra and some glands as well, but these are rather rare. So again, look at the surface. You see many, many different layers, and these are columnar. So we know these are stratified columnar. Here you see many layers of cells, but these are cuboidal shaped, so we call them stratified cuboidal. Right. Finally, there is a different class called transitional epithelium, also called urothelium. And this is, again, a different type because it's unique to the urinary tract, and it kind of transitions between two different formations. There could be a collapsed shape. When they're rounder, the cells look rounder, but they can transition to a more distended or flat shape when they're stretched. So transitional epithelium, or the urothelium, is found in the urinary tract and the bladder, and this allows for it to fill. Right. So when our bladder is filled, then our cells would be more distended and flat. When our bladder is not filled, then the cells of the transitional epithelium, or the urothelium, will be more round. So again, these transition depending on the state of stretching. And again, that's an important structure to function connection, right? Because of this structure, it allows the ureter and the bladder to function. So to review, we said that there's simple, pseudostratified, or stratified. The cell shapes could be squamous, cuboidal, or columnar. And then we mentioned this other type called transitional epithelium, or urothelium. The book doesn't use the word transitional thelium, it just mentions urothelium, but most textbooks would say transitional epithelium. So pause here and answer this question. So the answer is B, right? We need pseudostratified ciliated columnar epithelium to line the upper respiratory tract so we can get rid of any pathogens or dust and trap them in mucus and then propel them toward the digestive tract. Which of the following cell types lines internal surfaces of blood vessels? So pause here. So you should be thinking, what do blood vessels need to do? They need to rapidly exchange gases, right? So that's for diffusion and filtration. So we're going to need simple squamous cells for that, right? And those form the endothelium, the internal lining of blood vessels. The epidermis consists of, so pause here. The epidermis is skin, and that is composed of D, stratified, keratinized, squamous epithelium. So here's a book, here's a little video that's included in the ebook that I'd like you to review. This just talks about a brief overview of all the different classes So it tells you the functions. So we have transitional, pseudostratified, keratinized versus non. And then this talks about some of these junctions that we'll mention later in the lecture. So that marks the end of our talk on epithelial tissues. Now we can talk about connective tissues. Connective tissues are the most abundant and widely distributed tissues throughout the body. And unlike epithelial tissues, connective tissues are never found on exposed surfaces. Connective tissues have the ability to regenerate. And in general, while connective tissue is very diverse, they have a lot of differences between the different types. All connective tissues have three basic ingredients. They have ground substance, they have protein fibers, and they have some cells. And generally speaking, connective tissue has very few cells compared to the amount of extracellular matrix. What that means is the stuff outside of the cells is extracellular. So the matrix consists of protein fibers and ground substance. Right? So most of connective tissue is ground substance and protein fibers with a couple of cells embedded in it. Right? So here we have an image of loose connective tissue. We have very few cells, right? Like only a couple of cells, but we see protein fibers, and all the stuff that it's embedded in, the fluid, is ground substance. So much of the extracellular matrix is ground substance. Depending on the type of connective tissue, it'll determine what texture the ground substance is. But regardless, it's a complex mixture of carbohydrates, proteins, and other molecules. Very frequently, we see an abundance of glycosaminoglycans, proteoglycans, and adhesive glycoproteins. So again, these are a bunch of carbohydrates and proteins that are in the jelly-like the ground substance, and these help keep the cells embedded in the tissue. So it's like the glue almost that keeps cells stuck in the tissue. The extracellular matrix varies in texture. So in the case of fat, it's very squishy, but you also have like tendons and cartilage are also connective tissue. Those are more rubbery. Bone is a type of connective tissue that's more rigid. In blood, the extracellular matrix is a liquid. So again, we have different consistencies of extracellular matrix. But the cells are what actually secrete the proteins that make up the matrix. Specifically, fibroblasts are cells that secrete the protein collagen, which is one of the most abundant proteins in the body. Collagen is tough and flexible, so that's what provides our tissues with so much structure. So fibroblasts make collagen. This is a picture of a fibroblast, and here is a pink collagen fiber. So connective tissue has many, many functions. Binding of organs, for example, so tendons bind muscle to bone, ligaments bind bone to bone, fat keeps tissues in place, fibrous tissue helps keep our skin attached to our underlying muscles. So connective tissues connect things. They help us with structure and support. For example, bones support the body, cartilage supports the ears and the nose and the trachea. Connective tissue helps us with protection. So bones, like our ribs, protect our heart and protect our thoracic cavity. We have our fat protects the lining of our internal organs, protects our kind of cushions all of our organs so they don't move around a lot or aren't susceptible to damage. Connective tissue is also responsible for movement and connection, right? So bones and the tendons and the ligaments that help us move are connective tissue. They're also responsible for storage. So fat stores energy, right? It's our major energy reserve. Bone is how we store calcium and phosphorus that we can use when we need it. Connective tissue has a transport function because blood helps transport gases throughout the body. Right? It also helps eliminate waste. There's an immune function, right? So connective tissues have cells that can attack foreign invaders, right? So the bone bone marrow has these immune cells. And finally, in children and in babies, some of the fat has some heat productive function. So it can produce heat. So connective tissue, again, has many diverse functions. So I'd like you to be familiar with a connective tissue disorder called Marfan syndrome. And you can have Marfan syndrome if there's just one mutated gene called FBN1 on chromosome 15, because this one gene codes for a very important connective tissue protein. So without this protein, you have a lot of different unrelated defects because everything requires connective tissue. So for example, people with Marfan syndrome have disproportionately long appendages. They have a weakened aorta. And a lot of times it's an enlargement of the aorta, and they have very poor eyesight. They could also have breathing issues. They can have stretch marks in skin. So all these different organs require these connective tissue proteins. So just by missing one protein, you could have consequences throughout the body. That's how widespread connective tissue is. So let's discuss areolar connective tissue first. Areolar connective tissue is a type of loose connective tissue because it has very few cells and proteins. It looks like there's a lot of empty space. And areolar connective tissue is the most abundant in the body. It's found in tissues from almost every single part of the body. It surrounds our blood vessels and nerves, gets into the muscles, tendons, and nearly every epithelium rests on a layer of areolar tissue. And the areolar tissue has blood vessels that can provide the epithelium with nutrition and waste removal. Right. So like I said, it's found in the skin. Areolar connective tissue is a little stretchy in texture. It has some elastic fibers and collagen fibers. Like I mentioned, it serves as the supporting framework for internal organs, nerves, and blood vessels. Because the areolar tissue is so loose and has so much fluid-filled space, leukocytes, which are types of immune cells, can kind of travel about freely and then can find and destroy any pathogens. Right? So it's good that it's loose, so our immune cells can migrate around and eat up anything that's threatening to us. Reticular tissue forms the supporting meshwork for soft organs like our spleen, liver, and lymph nodes. So it's like, again, it forms a framework or a meshwork, almost like a sponge. And these spaces are filled with white blood cells called leukocytes or reticulocytes. So we see reticular tissue where we need to filter things out, like in the lymph nodes, spleen, and also the bone marrow. So now let's talk about dense connective tissues. These are different from loose connective tissues because these are dense with proteins. Right? When we looked at loose connective tissue like areolar tissue, those had very, very few proteins. Right? It was a lot more space. But dense connective tissue is dense with proteins. And there's two kinds of dense connective tissue. There's dense regular and dense irregular. Okay? So dense regular connective tissue has very few fibroblast cells and an abundance of parallel, tightly packed collagen protein bundles. So let me explain. They have a lot of collagen fibers, and these collagen fibers are arranged in bundles that are arranged parallel to each other. Right? So the parallel arrangement of fibers is very, very important because we see dense regular connective tissue in tendons and ligaments. And tendons and ligaments need to pull in very predictable directions. So we need these collagen fibers to be parallel to each other so we can have some directionality in our movement. So there's not only a lot of strength because of the collagen, but the parallel nature of the collagen bundles allows for flexibility. Right? So it's like a rope made of collagen fibers, but there's flexibility in that strength, right? A rope is very strong, but you can also move it around in certain directions. So again, we see dense regular connective tissue in ligaments, which connect bone to bone, and tendons, which connect bone to muscle. So we need a lot of strength here. We need flexibility. So that's where we need dense, regular connective tissue. Dense, irregular tissue has the collagen bundles arranged irregularly. So this is a little different. So it's thick bundles of collagen, very little room for cells, but the collagen bundles run in very random directions. And this is good for stress reduction. This could help us resist any stress. We see this in the dermis, which is just deep to the epidermis, and also in joint capsules. So when you think about stress resistance, right, we need a lot of, I'll show you a picture of this is the dense irregular. This is a lot more, it's a lot better to resist stress, but this is better for flexibility and movement. So we have tendons and ligaments, obviously, are moving, right? Moving bone to bone or bone to muscle. So we need to have this flexibility of the dense regular. But in our dermis, we want more structure and support and less movement. So dense irregular is perfectly fine with the random orientation of the collagen bundles. Elastic connective tissue has an abundance of regularly arranged elastin fibers. And elastin fibers are stretchy. They can recoil like a rubber band. So these elastic connective tissue will be important in things where we need elasticity. So think about the lungs, right? We need our lungs to expand. Our vocal cords need to expand and contract. Our air passageways and our artery walls, again, are always moving. We need elasticity. So some of these tissues have an abundance of elastin fibers, which allow for this recoiling. So now let's talk about adipose tissue, or fat. Right? Fat is consisting of mostly adipocytes, which are the fat cells that store fat droplets. So adipocytes are fat cells. You can see that the nuclei kind of get pressed against the membrane. The nuclei can barely be visible because they're so filled with fat. So the fat droplet inside of the adipocyte presses against the membrane. It almost like squeezes the nucleus to the very boundary. So each cell is filled with a fat droplet, right? And that fat droplet, again, is consisting of triglycerides. We need fat to cushion and insulate our organs, but also for long-term energy storage. Right? So it's good for protection, insulation, and long-term energy storage. We have adipose tissue deep to the skin in the hypodermis, and also around all of our internal organs to cushion them. So now let's talk about cartilage. So you could kind of feel your outer ear right now, your external ear or the tip of your nose, and you can feel cartilage. And it's relatively stiff connective tissue with a flexible rubbery matrix. Right? So it's a more firm gelatinous matrix, right? This time we have special cells called chondrocytes. Chondro always means cartilage, living in lacunae. So this sounds like a little weird, right? So these lacunae are these little lakes, little spaces within this gelatinous matrix that allows for these cells to live inside. So each of these is a lacuna. It's a little space that allows cells to live inside. They're fluid-filled pockets, right, within the matrix where the cells can live. The cells can't live in the gelatinous part, the non-living part. Cartilage usually lacks blood vessels, and that means that cellular processes are slower because you can't get oxygen in and wastes out as fast. Cartilage is very important for protection, support, and cushioning. There are 3 different types of cartilage that has differences in the kinds of fibers that they have present. Cartilage is covered with perichondrium that is made of dense, irregular connective tissue. So let's talk about the different types of cartilage. The first is called hyaline cartilage. And hyaline cartilage forms the trachea, it forms the larynx, and parts of the nose. It also attaches the ribs to the sternum. So costal cartilage is made of hyaline cartilage. Hyaline cartilage is also found at the protective coverings of bones at movable joints. We call this articular cartilage. We'll talk a lot more about that in the skeletal system. So we see articular cartilage at the end of movable joints. We see it in the trachea, we see it in the nose, and we see it in the costal cartilage that attaches the ribs to our sternum. Right. Most of the embryonic skeleton starts as hyaline cartilage, and it's then replaced with bone. Right, so in general, so hyaline cartilage is the most abundant. Elastic cartilage has elastin fibers. So it has like a web-like mesh amid the lacunae. And this we see in our external ear and in the epiglottis. Finally, we have fibrocartilage, which has a lot of collagen fibers. And this we see in our in the intervertebral discs. So in between our vertebrae, we need to have a lot of shock absorption. So that collagen, along with the cartilage, provides a lot of strength and support. So as we're moving around and walking, we have enough support between our vertebral column bones. So again, it's really important to absorb any shock. So fibrocartilage, we also see in the pubic symphysis, which is the joint between the two halves of the pelvic girdle. So it resists compression and absorbs shock. That's the main purpose of fibrocartilage. Right? Whereas elastic cartilage was more flexible and elastic. So what makes dense connective tissue so dense? I can pause here. So the answer to this question is B. An abundance of proteins is what makes dense connective tissue so dense. What type of connective tissue enables leukocytes to migrate around? So pause here. And the answer is A. Areolar connective tissue is loose enough, has enough space, so that white blood cells and other immune cells can migrate around and get rid of any pathogens that may threaten us. So other types of connective tissue include bone. It is the hardest and most rigid of all the connective tissue due to the calcium salt in its matrix, but also has a lot of collagen. I will talk a lot about bone in chapter 7, 8, and 9. It provides a lot of rigidity and strength to the body. Blood is also a connective tissue that consists of numerous formed elements, different cells, in a liquid matrix, a plasma. And blood has a vital role in the transportation of materials and gases throughout the body. So blood and bone are both connective tissues. This is a good time to watch this video contained in the ebook that again just talks about all different kinds of connective tissues, compares and contrasts them, and again gives you a good overview of what you need to know. I'm not going to test you on blood and bone much because you're going to learn about that in later classes. So recall that there are 4 types of tissue. We spoke about epithelial tissues already. We spoke about connective tissues already. Now the next category is nervous tissue. And nervous tissue is found in the brain, spinal cord, and nerves. There are 2 main types of cells in nervous tissue. There are neurons, which can conduct electrical signals, and neuroglia, which support the neurons. The neurons are the actual functional units of nervous tissue that possess a cell body, and they have processes that can connect to other neurons. Neurons respond to environmental changes by making nerve impulses, and they, again, can transmit different messages throughout the body. And the neuroglia are supporting cells that help nourish, insulate, and protect the neurons. The 4th category of tissue is muscle tissue. So we said epithelial, connective, nervous, and muscle. Muscular tissue consists of elongated cells that are specialized to contract when they're stimulated. Right? So their primary job is to exert physical force on other tissues and organs. Muscles also create movements internally, like digestion, waste elimination, speech, blood circulation. And we have different kinds of muscles. We have 3 different types of muscle specifically. We have cardiac muscle in our heart. This is involuntarily controlled. We have skeletal muscles that are voluntarily controlled. And then in our gut and our other internal organs, we have smooth muscle, which are involuntarily controlled. And again, we'll talk all about muscle in later chapters. So let's talk about cell junctions next. So cells within tissues need to stick together. Many of them need to also communicate. And cell membranes are kind of in the way of free communication. And if you had cells not attached to each other, then anytime they would contract, for example, they would just fall apart. So we need to have junctions. Cellular junctions allow cells to communicate very rapidly and allows them to stick together. Animal cells can communicate through gap junctions. So a gap junction is almost like a little protein tunnel that can connect two adjacent cells and attach it. It makes a membrane tunnel. So materials can move from one cell to the next very, very quickly. So in cardiac muscle and smooth muscle, gap junctions allow for electrical excitation to pass directly from cell to cell so they can contract in unison. That's very, very important. So ions, glucose, amino acids can go directly from one cytoplasm to the next through gap junctions. Allows for rapid communication and transmission of materials. Animal cells need to stick together. They do so through tight junctions. So tight junctions are what connect epithelial cells and join them tightly to neighboring cells. So 2 plasma membranes of 2 adjacent cells come very closely together, and they're fused by tight junctions, almost like sewing machine stitches. This allows for a leak-proof barrier. So this is really important in the stomach and intestines. Tight junctions prevent any digestive juices from seeping between the epithelial cells and digesting the underlying connective tissue. So all those epithelial cells need to be tightly attached to each other, so no acid can get past it. Desmosomes are a type of adhesion junction that can keep adjacent cells attached by linking their internal cytoskeletons. So these are almost like a clothing snap on a pair of jeans. They can snap 2 cells together by attaching their cytoskeletons. What happens is the desmosomes can connect intermediate filaments of one cell to the intermediate filaments of another cell. Remember that intermediate filaments are types of cytoskeletal proteins that provide structure. So in this way, we're linking one cytoskeleton to another through these proteins called desmosomes. So in this image, you can see these are tight junctions, here's desmosomes, and gap junctions. And then this is the image of the book that shows all 3 types. Tight junctions up here prevent passage of materials. We have desmosomes, keep cells tightly attached to each other. And again, we need these, very important because we don't want cells to pull apart from each other. And this is what was deficient. There was an issue with this in that disease, epidermolysis bullosa simplex. Remember, those skin cells kind of just tore apart because there was nothing keeping them attached. And those gap junctions allow for rapid communication and transport of molecules. So now let's talk about epithelial membranes. Body membranes are thin sheets of tissue that line body cavities, that can cover the surfaces of organs, and can separate tissues. Right, so body membranes line cavities, cover surfaces, or separate tissues from one another, or separate organs from one another. Epithelial membranes specifically are composed of epithelial tissue overlaying loose connective tissue. So again, epithelial membranes, a membrane equals the epithelial tissue plus the underlying loose connective tissue. Sometimes the membrane can also include a muscle layer beneath it. But we'll talk about three kinds of epithelial membranes. The first is a mucous membrane, then a serous membrane, and finally the cutaneous membrane. So these are three types of epithelial membranes. Let's talk about the mucous membrane first. So a mucous membrane is only found on the internal surfaces, right? And it lines the passages that open to the exterior environment. So any tube that is exposed to the external environment has to have a mucous membrane. Right? So the digestive tract, the respiratory tract, the urinary tract, and the reproductive tract all have mucous membranes. A mucous membrane consists of pseudostratified columnar epithelium with cilia above areolar connective tissue. We call the areolar tissue the lamina propria. That's the specific name that we call the areolar connective tissue layer. Sometimes there is a smooth muscle layer beneath it as well in the mucous membrane, called the muscularis mucosae. Of course, since it's a mucous membrane, we're going to need lots of goblet cells to make mucus. Right? And again, remember, anything exposed to the outside can have pathogens or dust, so we need to make the sticky mucus to trap them and to propel them with the cilia toward the digestive system, where they're not a threat. Right? It also helps keeps our cells moist and our surfaces lubricated, which is important to avoid any friction. So that's mucous membranes. The next kind of membrane we'll talk about are serous membranes, or the serosa. And serous membranes are composed of simple squamous epithelial cells above a thin layer of loose connective tissue. So again, before, mucous membranes had pseudostratified columnar epithelium, but in serous membranes we have simple squamous epithelial cells. So again, it's different. The epithelium of a serous membrane is called the mesothelium. The mesothelium. So the epithelial component of a serous membrane is called the mesothelium. And the serous membranes produce a watery serous fluid, and they line the insides of body cavities that could basically make sure that they don't have too much friction as they expand. We don't want organs rubbing past each other. So serous membranes allow for the serous fluid to kind of lubricate and reduce friction. So, of course, all membranes are protective. They help protect our organs, but the serous membranes secrete a serous fluid in between two membranes to reduce any friction. And it's a little hard to illustrate here, but if this is one serous membrane, there would be another right above it, like a mirror image of this. And there's serous fluid in between the two membranes. And again, the serous fluid allows for more flexibility and lubrication. So there are three kinds of serous membranes that you should be familiar with. We'll talk about first the pleural membranes, which line the thoracic cavity. So the pleural membranes line the thoracic cavity to protect the lungs. And again, it allows for the expansion of the lungs. So instead of, when the lungs expand, they have enough lubrication to expand, right? They're not pressing against anything. They're not going to rub against anything and cause any abrasion because they're protected by two serous membranes. The membrane that is closer to the lungs is called the visceral pleura. The one that is more superficial is called the parietal pleura. But it's a double membrane. Right? So the pleural membrane consists of the parietal and the visceral pleura. And then in between, we have that pleural fluid, the serous fluid. Specifically, in the case of the pleural membrane, we call it pleural fluid. The pericardium envelops the heart. So it protects the heart and provides lubrication to allow the heart to move and beat within its cavity. So again, we have the parietal pericardium and the visceral pericardium. And in between, there is pericardial fluid. Right? That serous fluid actually comes from blood. So that's where it's derived from blood serum. That's where serous membrane comes from. Finally, we have the peritoneum, which lines the abdominal cavity walls and the surfaces of abdominal organs. So the peritoneum provides protection and lubrication for the abdominal cavity and allows our digestive organs like our intestines to move and contract and expand without rubbing against each other and causing too much friction. So again, we have this double membrane. We have the visceral and the parietal, right? The visceral is deeper. And then in between, we have this fluid in between the double membrane. So those are serous membranes. We spoke about mucous membranes, serous membranes, and finally, cutaneous membranes is very simple. The cutaneous membrane is simply the skin covering the body. So the cutaneous membrane will be discussed in detail in Chapter 6, the integumentary system. But the cutaneous membrane consists of stratified squamous epithelium, right, that is keratinized above a layer of connective tissue called dermis. So the cutaneous membrane, again, all membranes are epithelial plus connective tissue. So in the case of a cutaneous membrane, we have stratified squamous keratinized epithelium above connective tissue. Right? Specifically, this connective tissue is dermis. And we'll see, and if you remember, the dermis, right, is dense with proteins. Right? It's not loose areolar connective tissue. It's very dense. So which of the following is not lined with serous membranes? Let's pause here. And the answer is the respiratory tract, right, is lined with pseudostratified columnar ciliated epithelium. So you're not going to be tested on this, but connective tissue membranes are also found in the body to protect and lubricate internal organs. So examples are the meninges of the brain and spinal cord. Right? The meninges are connective tissue membranes that protect the brain and spinal cord. Periosteum covers the outer layer of bones. And synovial membranes protect and lubricate joints. So these are formed only of connective tissue, not of epithelial tissue at all. But again, these are membranes because they help protect internal organs. And again, we're not going to talk too much about these in this class. So that is the end of part 1. We will continue with part 2, which will talk more about human development and stem cells.